Hello everyone! Today we are going to talk about the first section in Chapter 28 on regulation of gene expression. So the first section has to do with genome organization, and these are the concepts that we will cover. So these are the concepts that are covered in the section. An organism's complexity increases with gene number, but not necessarily with genome size. Ribosomal RNA and transfer RNA genes occur in clusters. We already saw some of that when we talked about in the RNA chapter. And eukaryotic genomes contain segments of repetitive DNA. So first, let's talk about something called the C-value. The C-value describes the rough correlation between the organism's quantity of genetic material and organismal complexity. So I have some pictures to show you in the next couple of slides. The C-value paradox, though, indicates this isn't always true. It mostly is, but there are exceptions. The other thing that's interesting is that only a small portion, and I mean a really small portion of the human genome encodes proteins, 1.2%. So humans encode about 21,000 genes, which doesn't seem like a lot, but we can increase the complexity by alternative splicing and post-translational processing, which will increase the repertoire of the proteins that we have. So this figure right here shows on the y-axis, this is the approximate number of cell types. So bacteria, of course, are single-celled organisms, and mammals are at the top with having the greatest number of cell types. And if you look at the haplogenome size, you would expect mammals to be right up here in this corner, but we're not. You wouldn't expect lungfishes or salamanders or any of these plants to have a greater genome size than humans, but they do. So that's part of the C-value paradox. This is shown here. So this figure, this table in our book shows the model organisms. So these are the kinds of organisms that people use in labs. So research scientists use these in labs for different kinds of genetic and biochemistry and molecular biology experiments. So we of course want to study humans, but if you're looking at what happens over generations, you might want something that produces generations a little faster than humans. So I was happy to see Haemophilus influenza up here. We did not work with this bacterium in our biochemistry laboratory course, but we did work with its carbonic anhydrase gene. So E. coli, yeast, C. elegans, this rice species, Arabidopsis, Drosophila, mice, humans, these are the kinds of organisms that geneticists will study. And there is a rough correlation between the genome size and the complexity of the organism. If you look at the number of genes, though, here, especially if you look at humans, you might find it surprising that the rice and the Arabidopsis express more genes than we do. But again, we can explain that by the use of the alternative splicing and post-translational processing. That will give us a wider variety of proteins with lots of different functions. This is an admittedly extreme example, but this gene right here in Drosophila has all of these exons, and if you look at all of the possible splicing patterns that you could have, this shows just one out of 38,016 possible splicing patterns. Now, people who work with Drosophila have studied this, and only a fraction of all of these different possibilities have been actually occur. So just because there's mathematically that number of possibilities, that doesn't mean physiologically you get that many. But it's still, the point is made that you can take this one gene and you can make lots and lots of different proteins from it. So the way that this works is that splicing can be regulated either by repressors or by activators. So if we have our pre-messenger RNA transcript, if nothing is regulated, this intron that's shown in yellow will be spliced out, and we will get our mature messenger RNA right here. But if we have a repressor, the repressor protein can block the 3' splice site, and what happens is splicing doesn't occur. And now what was an intron becomes an exon and becomes a translated part of this messenger RNA transcript. We can also have activators that do the opposite. In the absence of the activators, splicing does not occur. But if the activator is present, the activator can activate that splice site, and now this yellow portion here becomes an intron and is spliced out. So the presence of repressors and activators can control whether splicing occurs or not, and that will produce a different messenger RNA transcript, which produces a different protein. So here's an example from Drosophila. So in fruit flies, this ultimately will explain how these DSX proteins are produced. So a female DSX protein and a male DSX protein have different structures and different functions, and these proteins go on to carry out other activities in which they specify the characteristics of a female or a male fly. So in here, the females are shown in pink and the males are shown in blue. So the female produces this SXL protein, so that must get started somehow, but there's a feedback loop where the SXL protein acts as a repressor of splicing. So in the female, what happens is that this exon right here is skipped, and you go from 2 to 4, and then you produce this SXL protein. So this protein right here is what you get in males. So in males, this skipping doesn't occur, and this this intron, this number 3 exon is present. So this isn't SXL, it's something else in the males. But the SXL in females then acts as a repressor for the TRA protein and also leads to exon skipping. So in the female, you go from 1 to 3, and then this is the female TRA protein. That doesn't happen in males, so you get a different kind of protein. The TRA protein then, along with RBP1 and TRA2, acts as an activator, and this activates splicing here. So for the females, you get the DSX protein that looks like this, whereas in males, the males actually skip over this because they don't have the activator. So they produce a different DSX male protein. So this shows this complicated sequence of events that produce the female specific and the male specific proteins that lead to the characteristics of female and male flies. It's interesting to me, if you look at this pie chart, that 41.7% of the genes are, their molecular function is unknown. It seems like we should have known all of it by this point. So we do know, if you look at the human genome, so the human genome was sequenced about 20 years ago, and a lot of these functions have been mapped to the genome, but there are lots of genes with unknown function. We don't know what they do. So that'll be interesting to find out. So we talked about how prokaryotic cells have genes cluster in operons, but eukaryotic genes, even though they don't have operons, have some kind of similar organization where gene clusters will occur. And we already saw this in the RNA chapter, where we saw that you could generate one huge ribosomal RNA transcript, and then you could process it to get the 18S and the 5.8S and the 28S ribosomal RNA molecules that make up the ribosome. So protein coding genes almost never occur in multiple copies, because if you have a messenger RNA transcript, you can use that same transcript and get lots and lots of protein. An exception is histone proteins. There are lots of genes for histone proteins because during cell division, the cell has to ramp up its production of histone proteins, so we need lots of copies of that. So this looks almost sort of operon-like. Remember when we talked about the structure of histones in a previous chapter, that we studied the functions of these histone proteins, and they are located very close to each other in these repeating units. For some reason, our authors decided to include these are sea urchins, so they are related. But anyway, and these are flies. I'm not sure what this is, but this shows the location of the histone proteins. They're very close to each other, so that means that you can regulate the production of these proteins very easily when it comes to cell division. So this is an interesting example from hemoglobin. Various genes are transcribed at various developmental stages. So here we have our A-like genes are on chromosome 16, and I should say alpha-like genes, sorry, alpha-like genes on chromosome 16, beta-like genes on chromosome 11. And um, there are different genes that are expressed at different points in development. So the first hemoglobin made by the human embryo, and this is really early before you even really have a circulatory system. So this is zeta 2 epsilon 2. So the zeta is sort of an alpha-like gene, and then the epsilon is right here. So that's made in the first 8 weeks. And after 8 weeks, what we've described as fetal hemoglobin is made next. And we've talked about fetal hemoglobin. So it's alpha 2, so two alpha chains and two gamma chains. And these are the two gamma chains. They're the same except that at position 136, this gene has a glycine and that has an alanine, but that apparently does not impact its function very much. That's a conservative substitution. In Biochem 1, we talked about how fetal hemoglobin has a higher oxygen binding affinity than maternal hemoglobin, and that helps oxygen transfer from the placenta or occur between the maternal and the fetal circulation. So adult hemoglobin, so adults stop making the fetal hemoglobin, but never completely stop. About 1% of our hemoglobin is fetal hemoglobin. We also have 2% of this variant called alpha 2 delta 2, but 97% of our hemoglobin is the alpha 2 beta 2 molecule, the tetramer that we talked a lot about in Biochem 1. You might also notice from these figures that there are pseudogenes. So our authors describe these as non-transcribed relics of ancient gene duplication. So some kind of holdover from evolution. So this graph right here shows the progression of these hemoglobins and how they're made. So here's birth right here. So this is a 40-week gestation period right here. So the first hemoglobin that's made is the alpha 2 zeta 2, and then fairly early on you switch over from alpha 2 epsilon 2, and then here for a huge part of the pregnancy, the fetus makes the gamma 2, the fetal hemoglobin. Again, this has a higher oxygen binding affinity than maternal hemoglobin. But the fetus, especially when you get close to birth, the production of the gamma chain drops and the beta chain ramps up. But this is in the first 48 weeks of the neonate, postnatal age. You can see that really up until 12 weeks or so, the newborn baby is still making a lot of fetal hemoglobin. And again, that fetal hemoglobin never goes away. It goes down to about 1%, but some of it's still there. And there's the delta right there. But most of our hemoglobin, again, for the fetus, say from 12 weeks on, is the alpha 2 beta 2 hemoglobin that we studied in Biochem 1. So if we look at the nucleotide content of the human genome, it's maybe a little puzzling, at least for me, because I'm not a geneticist, but there are lines, which are long interspersed nuclear elements. Our book authors call this molecular parasites. They're transcriptionally inactive. Signs are short interspersed nuclear elements. The most common example is the ALU family. So ALU1 is a restriction enzyme, and apparently there are lots of ALU restriction sites here. So if these are repetitive sequences and maybe not, quote unquote useful, whereas these are unique sequence right here. So we have non-repetitive DNA. Here are introns, and then way over here, this little tiny bit right here are the protein coding regions. So I find this fascinating. So what you can do is, if you look in those portions of the DNA that aren't transcribed, you would expect evolution to occur more quickly there because there aren't any consequences to any changes in DNA sequence like there would be if you had a change in hemoglobin, for example, which has have to happen with sickle cell or any of the other diseases that we talked about. That if you have a change in the DNA sequence, you'll change the protein sequence, and this could have either mild or maybe dire consequences for the organism. But if you're in one of those places that is not transcribed, you can then take all of the peoples of the world in different places around the world and construct this evolutionary tree where we can go back in time and imagine what our common ancestor may have looked like. So that is the end of section 1, and here are my references, and see you next time.